In this session we look at similarity measures and non-metric multidimensional scaling. First we set the working directory and then we load the library vegan, which we use to calculate all the functions of which we use for the calculation of the different association measures. As discussed in the lecture, there are different libraries that would provide you with these functions. So in case you don't find all the association measures you want to use, then other functions, other packages may provide these. We do these for an example matrix with 6 species that have been observed at 5 sites. So we create this matrix. We see that the species are in the columns, the different species, and rows represent the different sites, and we see that the abundances of the different species vary between the sites. So on some sites we have almost 200 individuals observed, whereas in other sites only 30. And the aim is now to calculate the different association measures, such as the Bray-Curtis, distance, Euclidean, and Jacquard distance or dissimilarities, and compare the results for those, and then check what is the relationship of the highest to lowest dissimilarity in the matrix, and you should calculate this using the vectors function. That's not particularly complicated. You just provide the data set to this function and the dissimilarity measure you want to use, or the distance measure. And you should check this out yourself by looking into the help of the vectors function. And in the second step, you should scale the matrix, or standardize the data before rerunning the analysis with the association measures. So you first standardize the data using the decostand function and standardize by the maximum, and then you recalculate the dissimilarities and distances and check whether the standardization has led to any changes in the results for the association measures. So that's something that you can do yourself. So the implementation in R should be relatively straightforward. So explore the effects of different association measures and of standardization. The solution will be provided to you in a couple of weeks after this session has been done in the course. So we move on to non-metric multidimensional scaling. Remember that that is a kind of ordination method that is not based on eigenvalues. And to infer how many dimensions we need, we can do this based on the goodness of fit measure, the stress that is produced, or is the stress that is included in ordination. Now we know that the initial configuration is somehow randomly produced and may yield 2 different end results. So for finding the optimal number of dimensions, we randomly replicate each dimensional step 1 time when we want to look at the resulting stress level. And this is done or automated in this function provided here. We don't go into details of this function, so we basically run, this is what you find here in the core of this function, meta NMDS function, which is the function that has been presented in the lecture. that does the multidimensional scaling and a lot of other data preparation steps and so on. So it's an easy to use function because it does several steps for you that you need. And this is run 10 times, so it's replicated and the results afterwards can be plotted. So you just have to execute or copy and paste this function. The arguments that you need to set for this function is, on the one hand, the data matrix, then the maximum number of dimensions you want to check, and the distance measure that should be used. So we use the dune vegetation data set. So this data set has been produced in a study where the vegetation or the species occurring in dunes has been analyzed. And you see here that when we first look at the species data, we see that the maximum abundance of each of the individual species, the maximum is much higher, it's in the same order of magnitude. There are no strong differences between the different species. So we don't need any standardization of the data in this case. And then we run our function that produces the scree values for a maximum of 10 dimensions. Typically, we would only, so the non-metric multidimensional scaling makes only sense if we can visualize the data appropriately in 2 or 3 dimensions. Remember that otherwise we don't have information based on eigenvalues or variance partitioning between axes, so it becomes rather tedious to interpret the results of an NMDS if you would need 5 or 10 axes. So anyway, we run this up to 10 axes in this case. As distance measure, we use the Bray-Curtis distance because we have abundances. And we see that this runs a couple of seconds. Just wait for the results. So within the function, it's the NMDS algorithm is run 30 times anyway to find the best result. And then we plot the result. We need a helper function here. Don't go into details here. You can simply copy this if you need to apply this to your data. And we produce the plot. We have the red line indicating the stress level. So all stress values below are having stress values that are producing an ordination that has good or better fit or excellent fit. And we see each of these points here, although that's not clearly visible, is actually 10 times replicated. So it's 10 replicates plotted upon each other, but that's only visible in the case of one dimension, which means that unless we have one dimension, then if we have 2 or more dimensions, then the algorithm always yields to the same result. So the result is fairly robust, we could say. But even for one dimension, there's not much difference in the stress values. We could, depending on our aim, we could argue here that 2 dimensions almost yield a good description of the data, representation of the data, or if you use 3 dimensions, then this representation is actually quite reliable. So we proceed. We select 2 dimensions and fit the NMDS again. It's fairly quick. And then we look at the Shepherd diagram. So this provides us with a non-metric R square and the linear R square that's not displayed here, and shows us the relationship between the ordination distance and the observed dissimilarity. So the dissimilarity between the sides that would be between distances of two sides in the raw data and the distances that have been mapped in the ordination space. So we see that there's a fairly good relationship between both, although there is obviously some scatter. Then we prepare for plotting. Since we have many species, some species would be plotted above each other, have a similar position, so we decide to only plot those that occur more often in the data set. That's calculated if we calculate the column sums. So that's an alternative to what we did with the standardization for presence absence and then summing up in the previous script on RDA. And then we plot the NMDS result. We use here the special function orditorp for plotting, since this function provides us with more flexibility when we plot the species score. So, and now we see the relationship between the species depending on the different, on the first, for the two dimensions that we have used. So we could interpret that the closer species are, the more similar they are, and we have the different scales on the y-axis and x-axis. So generally the similarity between two species decreases based on 1 unit of distance. We could also display the sides. It would be, so here the species are obviously plotted as weighted averages across the sides. We could also fit environmental variables to this plot. These are the environmental variables, and we don't go into detail here on these variables. We just transform some of the variables from factors into numeric variables, assuming that they make sense. Here we combine some factors, so that is just done for this type of data set based on some explanation for this data that I found in literature. But that's nothing you need to worry about, that's just data preparation. And then we use the environmental fitting algorithm and fit the tune environmental variables to this data set. So what is basically done, this algorithm checks for correlations in the resulting ordination distances, the correlations between ordination distances and the environmental variables. When you call this algorithm, you see you obtain information here on the vectors and the relationship between the different NMDS axis, and the R square provides the results from the regression of variables against the ordination scores, and then the p-value, whether this is statistically significant. And you also obtain the centroids of our factors that we have in that we've used in fitting. So we can add these to the plot. Then we would see the centroids for the management type. So species that are closer to the centroids of these management type can be considered as occurring more often in this management form, whereas for the arrows, they are placed in the ordination plot regarding the gradients, the correlation of gradients. We see on the different axes, we see here correlations. So 0.9 would be the longest arrow, would be a correlation with the first dimension of 0.9. And this correlation would be regarded as linear. However, the response is not necessarily linear, and this could be visualized. So the arrows somehow suggest we have a linear response. And this can be done when we've used the ordeserve function for an individual variable. So we do this again. So here we see somehow that this arrow into one dimension is not quite true. We have more a wave-like surface. So the relationship, for example, along this gradient, there's a much stronger, shorter gradients. There's a much stronger change than along this line here in this plot. So we can fit again this first vector on the data that somehow suggests a linear response that is perpendicular along this vector, but this is not quite true. That's rather true on average. We see that response varies depending on where you are in the response surface. So here you have also an increase in moisture along this gradient, for example, that is not necessarily obvious from this arrow. Finally, we could explore 3 dimensions in R. This is done with the function vegan 3D that is provided in the package vegan 3D with the library. The function is called RGL. So we fit again the NMDS on 3 dimensions, and then we plot this again, and now we obtain a 3-dimensional representation of the ordination space. So we can move that and explore the 3rd dimension by moving and this visual representation. Yeah, in the following you should do an exercise using the legend data that we have used before in the course. And here's also an extra section that provides a warning example regarding interpretation of linear gradients in the data set, and that plotting of the data may actually provide you with some information, or should always be done to provide you with some feel of how this linear gradient really, if this linear gradient really exists in the data. So with that, we end the session on similarity measures and non-metric multidimensional scaling, and hopefully you have now learned the basics to apply these methods in your research, and in the future I welcome you to other courses.